Welcome to Life Science Note, friends. So today we'll be seeing about gram staining. Gram staining is the most widely used microbiological staining technique. It is also known as differential staining technique. The gram staining was discovered by Christian Grams in 1884, and hence the name was put forward as gram staining. This technique categorizes bacteria into two groups, gram positive and gram negative, depending on their gram's nature. Depending on their gram's nature, the stain used in this technique categorizes the bacteria into two groups, that is gram positive and gram negative. And this reaction is dependent on their cell wall composition. That we will see in the principle of gram staining further. So here I have made 4 columns in which first are the stains and the reagent that are used. The second one is function of that stain and a reagent, the reaction in gram positive bacteria, and the fourth column is the reaction that happens in gram negative bacteria. First stain which is used is crystal violet. Crystal violet here in gram staining is primary stain used. The function of this stain is a primary stain. So in the reaction in gram positive bacteria, violet color complex is formed, crystal violet. And in gram negative bacteria also the violet color complex is formed, CV, crystal violet color complex. The second is a mordant use. So what is mordant? First I'll write the name, which is grams iodine. The function of grams iodine is it acts as a mordant. So what is a mordant? Mordant is a substance which forms insoluble complexes and which is responsible for depositing the stain on the cell. Means if we used crystal violet to stain a bacteria, then if we added grams iodine after discarding the crystal violet, it fixes the stain in the bacterial cell. It here I'll suppose this is bacteria, we added crystal violet. Okay, we allow it to fix for a few minutes or so, and after that we added grams iodine. So in the cell wall here, in the cell wall of the bacteria, this crystal violet will be fixed depending on the composition of the cell wall. Depending on the composition of the cell wall, this mordant will fix the stain in the cell wall of the cell. So this mordant, what it does in gram positive bacteria, that it forms C crystal violet iodine complex. In gram negative also it forms a CVI complex, which is crystal violet iodine complex. So the one decolorizer here is used, which is alcohol. As we have said earlier, that it is a differential staining technique. So obviously there are two groups of bacteria, gram positive and gram negative. So the decolorizer is important. The alcohol acts as a decolorizer, which decolorizes the stain from one of the two groups. So in the gram positive bacteria, the CVI remains the same. We'll study this, why the CVI remains in gram positive bacteria the same. We'll study in the principle of gram staining. But in the gram negative bacteria, the CVI complex is removed. Means what? That the stain from here is completely removed. Now the fourth one. So now the fourth one we will be seeing is about safranin. Safranin is a stain, is the second stain used in gram staining. The function of safranin is it acts as a counter stain. counter means opposite. The reaction it shows in gram-positive bacteria is nothing but the CVI complex that remains the same. Why? We will see further in the principle of gram staining. Okay, the gram-positive bacteria remains violet here, but the gram-negative bacteria accept the color, the stain saffronin, and become pink in color. So we have seen the function of two stains, as well as the reaction it shows in gram-negative as well as gram-positive bacteria, and we have seen what is the action of grams iodine and alcohol on gram-positive and gram-negative bacteria. So let's move forward to the principle of gram staining. So now we will see the principle behind the gram staining. So these are the two cell walls of the bacteria. Okay, the cell wall of gram-negative bacteria and the cell wall of gram-positive bacteria. So as you can see here, the peptidoglycan layer is much thicker in gram-positive bacteria, whereas the peptidoglycan layer is thin in gram-negative bacteria, consisting of 1 to 10% only, and here consisting of 90%. Okay, whereas the lipids here are very less, in a lesser amount, like 1 to 10%, whereas the lipid constituting here is 90%. So now we have seen that what is the difference between the composition of gram-negative and gram-positive bacteria. Now we will see the actual principle behind it. As I have said earlier, that the structure of cell wall of organism determine whether the bacteria is gram-negative or gram-positive. So the CV, when we add the crystal violet in a group of bacteria, in a colony of bacteria, this dissociate into CV plus and CV minus ions. Okay, so till here we have seen that we have added a crystal violet into a sample, and what happens is they dissociate into CV plus, means crystal violet plus ions, and crystal violet minus ions. Okay, these ions penetrate through the cell wall. These ions, CV plus and minus ions, penetrate in the cell wall and then cell membrane of the cell. The CV plus ion interact with the negatively charged compounds in the cell wall of the bacteria. The negatively charged compound in the gram-positive bacteria is teichoic acid, whereas the negatively charged components in gram-negative bacteria is a lipopolysaccharide. So what happens is this CV plus ions interact with lipopolysaccharide and teichoic acid in gram-negative and in gram-positive. Okay, the CV plus ions interact with the negatively charged components of gram-positive and gram-negative cell of the bacteria and color them into purple color complex. The teichoic acid is negatively charged in the gram-positive and the lipopolysaccharide is negatively charged in gram-negative bacteria. The teichoic acid contain phosphate in their structure, so therefore they are negatively charged. When the iodine, it is a mordant, right? When the iodine is added, they fix the stain in the cell wall of the cell. Okay, iodine acts as a fixer that fixes this crystal violet in the cell wall of the cell. After what happens, this iodine interact with the CV plus ions and form CV plus iodine complex. then what happens is there is an addition of decolorizer, which may be alcohol or acetone. Mainly alcohol is used as a decolorizer. So when the decolorizer is added, some bacteria remain stained as purple while other decolorized themselves. When a decolorizer such as alcohol is added, it interacts with the lipids and dissolve this lipid layer. Dissolve this lipid layer. The alcohol dissolve the lipid layer. The percentage of lipid is higher in gram-negative bacteria, so it dissolves the lipid layer. So when the lipid layer is dissolved, obviously the stain is removed. The stain gets removed. So there is an ample space that the other counter stain can accept the stain. Here we have seen what the alcohol, what is the action of alcohol on gram negative. But on gram positive, what happens is, as it doesn't get decolorized because the lipid content is hardly 1 to 10%, so the pores get shrinked and the stain gets locked in gram positive bacteria. So they remain purple. When the counter stain is added, which is safranin or maybe basic friction, when the counter stain is added, this cell wall of the gram negative bacteria accept that counter stain and stain themselves pink in color. So this is how the principle of gram staining is. I hope you all understood. Thank you for watching my video. Please don't forget to like, share and subscribe to my channel. I'll see you in the next video. Thank you.